everyone. This is Dr. Ahmad, and today we are going to learn the next topic in our renal system, and this is going to be renal histology. We have already focused on renal anatomy, and this is going to be renal histology, and we'll focus on different type of cells and the general structure of kidneys and etc. So, what are the learning objectives here? First of all, in this video, we're going to learn about the gross kidney structure, just to review. We're going to learn about the renal lobules, nephrons, and renal corpuscle, glomerulus, and filtration apparatus. There are different portion of collecting ducts that we're also going to look at. We're also going to learn about macula densa, and juxtaglomerular apparatus, blood supply, vasa recta, and its origin, and understand the histology and functions of ureters, bladders, and urethra. So here we go. So what is the overview of the renal function? So all these structures, they come together to perform some major functions of the kidneys that includes the removal of toxins while simultaneously retaining water, salts, and proteins and amino acids. It will also be assisting in regulating the blood pressure and acid base balance of the body fluids, hormone production, for example erythropoietin, and assists in the production of vitamin D. So let's start with the general structure of the kidney. The very first portion is the renal cortex. It is the outermost part of the kidney, which is usually 1 centimeter in thickness. And the microscopic structures that are present in the cortex includes the Bowman's capsule, the proximal and distal tubules, and the upper portions of the collecting ducts. The next portion of the kidney is the renal medulla, and it is divided into the units known as the renal pyramids. So, 6 to 10 renal pyramids are separated by the columns of Burton, and the base of the pyramid is up against the cortex, while the apex is projecting towards the renal sinus, and the apex is called the papilla. The collecting tubules, they drain out of the papillae and into the minor calyces, and then they combine to form the major calyx. The microscopic structures present in the medulla are the loops of Henle and the collecting ducts. Then there is the collection system. The main function of this is to collect the newly formed urine and direct it to ureters. Each papilla drains into the minor calyx. 2 or 3 minor calyx, they combine to form the major calyx, and 2 or 3 major calyces combine to form renal pelvis. And finally, we have renal pelvis, which is a funnel-shaped structure which is continuous with the ureter and occupies most of the renal pelvis, and it transitions into the ureter at the uretero-pelvic junctions. So look at the general structure of the kidney again. So we have a cross section of the kidneys over here. However, then we have the microscopic structure that we're going to discuss that in detail. You see the renal hilum right here. You see the ureter on the posterior view. Then we have the renal nerve, which we're going to discuss in detail in later slides. We have the renal artery and the renal vein. The renal artery, it goes in and then it changes into the interlobar arteries. It ascends up and it makes the arcuate vessels. So it changes into these interlobar arteries, and then it changes the arcuate vessel. It then go up, arch around, and then it changes into the interlobular arteries. After the arcuate arteries, those interlobular arteries then further divides until they make the afferent arterioles right here. And these afferent arterioles then goes into make this tuft of capillaries, which is called glomerulus. And once they release, it's the efferent arteriole. It can either make a peritubular capillaries, or it can also make the vasa recta. We'll discuss that later. So functional unit of the kidney is the nephrons. And nephrons, they are made of 2 parts, renal corpuscle and the renal tubules. You see here, there are 2 types of nephrons that you can see. This is the cortex right here. This is the medulla. And you see here, this is the renal papilla, and this is the collecting system right here. So this is the cortical nephron, and this is the juxtamedullary nephrons. So cortical nephrons, they are located almost entirely in the cortex. However, there is a loop of Henle that has a small course in the medulla, and the juxtamedullary nephrons, they are located close to the corticomedullary junctions, and loop of Henle goes deep in the medulla. They're responsible for maintaining the high osmotic gradient within the medulla, and this is the high yield point right there. They're also responsible for the stronger urine concentration. Now, let's look at the different components of the nephrons in details. So, the very first component of the nephron is the renal corpuscle. So, it is the first major part, and it is the area where the blood plasma is filtered. So, the first part of the renal corpuscle is called the glomerular or the Bowman's capsule. It is a ball shaped structure which surrounds the tuft of arterial capillaries, and that tuft of arterial capillaries is called glomerular capillaries. And Bowman capsule or the glomerular capsule has two layers. The outer layer is called parietal layer, which is made up of the squamous epithelial cells, and it has the inner visceral layer, which is directly surrounding the glomerular capillaries and made from the specialized epithelial cells called podocytes. And what is the next part? It is the Bowman's capsule. It is a space between the parietal and visceral layer of the Bowman's capsule, which collects filtrate and drains it into the proximal convoluted tubules. So the blood vessels that come here, we discussed that before. We have seen that before. It is the afferent arteriole. Start with A, which means, I mean, if you look at the chronological order, A comes first, E comes after. So A means going in, E means going out. So afferent arteriole, it has a relatively large diameter and brings the blood to the glomerulus. And then there is a glomerulus. It is a tuft of small arterial capillaries between the afferent and efferent arterioles. And in this area, the filtration occurs. And the last part is the efferent arteriole, which has a smaller diameter and carries filtered blood out of the glomerulus. Important thing to remember is that the filtration occurs because of the high hydrostatic pressure in the glomerular capillaries, which is created by the large inlet of the afferent arterioles and a small outlet of the efferent arterioles. So if you look at the blood cells, it just stretch a little bit, so it makes the filtration easier. So let's look at the renal corpuscle right here. And this is the Bowman capsule right there. This is the afferent arteriole. You see a little bit of a bigger, a bigger artery. And it goes in there. This is the tuft of the capillaries. That is the glomerulus. And then there is an efferent arteriole. The blood leaves from there. You see that this is the parietal layer of the Bowman's capsule. The visceral layer of the Bowman capsule is made from these glomerulus. And you see these cells that is like covering the glomerulus. This is called podocytes. You can also appreciate the proximal convoluted tubule right there. So the next part is the filtration apparatus, and it's very interesting to see. It constitutes the main blood filter between the glomerular capillaries and the Bowman's capsule, and it has three layers. The very first layer is the endothelium of the capillary itself, which is fenestrated, which means it has a small gaps in it or small holes in it. The second one that covers the endothelium is the glomerular basement membrane, which is made from the negatively charged proteoglycan gel, and its function has the repulsion of the negatively charged molecules. So it is kind of acting as a molecular sieve. And the final part of the filtration apparatus is the podocytes. It is the epithelial cells of the visceral layer of the Bowman's capsule, and there are multiple foot processes that wraps around that vessel, which makes the filtration slits between the feet or the foot. And these slits are covered by the diaphragm, which is intercellular junction made from the multiple proteins. So it goes down very little detail on the molecular level to make sure that the filtration is done right. So this filtration apparatus does not allow the large molecules in the blood to be filtered out. So the blood cell itself, it does not leave unless there is a trauma or something else or another pathology present. The blood cell does not leave the capillary. And the plasma proteins like albumin, it does not allow that to be left through the membrane. However, the molecules which is less than 3 nanometer in size, like water, electrolytes, glucose, amino acids, fatty acids, and vitamins, they are filtered out. And this apparatus can be damaged by trauma or infection. So let's look at the cross section of the glomerulus or the filtration apparatus. So right here, this big lumen is basically the capillary lumen. You see the internal capillary endothelium, but it is fenestrated. Right here, you see little gaps around it. And then you see a single nuclear cell right there. After that, there's green a green structure that you see around it. That's the glomerular basement membrane in green color. And after that, there's a podocyte. So you see the podocyte, a big cell that is wrapping around the capillary in itself. And then it has these foot processes right here. So there's three different filtration places that the molecules has to traverse to go out into the Bowman's space to make sure that it's filtered out. Otherwise, it stays inside the capillary. The next part of the filtration is the renal tubules. This is the next part of the nephrons. It is a long tubule which adjusts the filtrate contents received from the renal corpuscle, and it finally drains through the papilla into the calyces. It has five different portions. So the very first portion is the proximal convoluted tubule. So it is majorly located in the cortex, and it is one of the major sites for the reabsorption. So if there is 180 liters of filtrate every day, it reabsorbs at least 65 to 75% of the filtrate contents, and the cells over here are extremely active. And that is because they are required to do the active transport, and because of that, they have a high amount of mitochondria. So if you stains with H&E, you see that dark pink color because of the high amount of mitochondria present, and there's a high rate of reabsorption that occurs here as well. And there is a reabsorption of electrolytes like sodium, chloride, potassium, calcium, magnesium, bicarbonate, phosphate that occurs here, and it also reabsorbs glucose, amino acids, and peptides. So this first part is very important in reabsorption because at least 65 to 75, that is like three-fourths of the filtrate that gets reabsorbed here. So the cells over here are very, very active, and the cells in the proximal convoluted tubules are lined with the simple cuboidal epithelium with prominent microvilli, and that is a differentiation point. If you're shown a slide and if you can appreciate the brush borders in there, then you will know for certain that it is the proximal convoluted tubules, and this will increase the surface area of the reabsorption. So their cells, they have their own walls, but then there is microvilli present, so that increases that surface area for the filtrate to be the electrolytes or different materials that need to be reabsorbed back in the blood. It happens through there. The next part is the proximal straight tubule, and it stains dark pink in H&E stains, and it is also a major site of our secretion of organic anions. For example, bile salts, urate, drugs, and organic cations, which is the creatinine, dopamine, and drugs. The third part is the loop of Henle, and it is located in both cortex and medulla. So if you remember from the previous slides, if the nephron is cortical, there's a small part of loop of Henle that goes into the medulla. However, if the nephron is juxtamedullary, then the loop of Henle actually traverses deep in the medulla, and depending on nephron. So divide it into thin descending and thin ascending limbs and a thick ascending limb as well. So the thin descending and thin ascending limbs are made from the simple squamous epithelium. They are involved in the passive transport of water and electrolytes, which establish the large osmotic gradient in the medulla. And it's a very important part. You'll learn more about this in the physiology lecture. The next part is the thick ascending limb, which is made from the cuboidal epithelium, and it is heavily involved in the active transport of electrolytes. Next part of the renal tubule is the distal convoluted tubule. It is lined with the simple cuboidal epithelium without the microvilli. So there is a difference. If you can appreciate the microvilli, it is made from the proximal convoluted tubule. That's what it belongs to. If there is no microvilli present, but there is like simple cuboidal epithelium, then it is distal convoluted tubules, and it stains pale pink in H&E stains due to low mitochondria, and it is responsible for the fine tuning of the components of the urine, and it is the last part of the nephron functional unit. The final part is the collecting duct. So it is lined with the simple cuboidal epithelium, multiple distal convoluted tubules from different nephrons. They drain here, and it is located in cortex, and it traverses deep into medulla. Now let's look at the blood supply of the nephrons. So up until now, we have established from renal artery all the way to efferent arterioles. So once the blood leaves the glomerulus, it enters into the efferent arterioles. But now, depending on the location of the nephron, it can either go into the peritubular capillaries or it can go into vasa recta. So peritubular capillaries, it supplies the cortical tubules. So any tubule system, renal tubule system, which is present inside the cortex, it is the blood goes through the peritubular capillaries. The proximal convoluted tubules and distal convoluted tubules are also getting a network from peritubular capillaries. However, if the nephron is the juxtamedullary nephron, then the capillary network that surrounds the loop of Henle in the medulla, it's vasa recta, and it is highly permeable to solute in water, and it is vital in maintaining the osmotic gradient in the medulla. So what happened to the blood after that? Blood from here drains into the interlobular veins. It go into the arcuate veins, interlobar vein, renal vein, and finally into inferior vena cava. So let's look at the difference between the peritubular capillaries and vasa recta through a diagram. So you see here, this is the cortical nephron, all of it. This is the juxtamedullary nephron. So you see the arcuate artery, arcuate vein going through here. You also appreciate the collecting duct, a big one, right there. So you see the cortical nephron, the efferent arteriole. Once it leaves there, it makes a network which is made from the peritubular capillaries. When you look at the juxtamedullary nephron, you see the efferent arteriole leaving, and it is making the vasa recta right here and going deep in there in the medulla. So next important part, which is also important with different other function, is the juxtaglomerular apparatus, and it is made from the three specialized cells in the close proximity, and their main objective is to maintain the blood pressure and the fluid homeostasis. So what are the three parts or three components? Are juxtaglomerular cells, macula densa, and mesangial cells. So the first part is the juxtaglomerular cells. They are enlarged smooth muscle cells, which is primarily present in the afferent arterioles. And once they are signaled because of hypovolemia or hypertension, they can secrete renin to activate the renin-angiotensin-aldosterone pathway or system, and they're able to dilate or constrict the arteriole and adjusting the pressure within the glomerulus. The second one is the macula densa, and they're closely spaced epithelial cells in the distal convoluted tubule, which are adjacent to the juxtaglomerular cells, and they're able to signal the juxtaglomerular cells to dilate or constrict arterioles, adjusting the GFR, glomerular filtration rate, in order to maintain the homeostasis. And they're also able to detect sodium concentration of the fluid in the distal convoluted tubule. And the last part of the juxtaglomerular apparatus is the mesangial cells. They are connected with the macula densa, and juxtaglomerular cells by the gap junctions, and they are present in the cleft between afferent and efferent arterioles. They're made from the flat and elongated cells, and their role is still unclear, but they may be working as a mediator between the macula densa and the juxtaglomerular cells. So let's look at the apparatus here. So it is the nephron right here. It is very closely integrated right here. So you see efferent arteriole right there, afferent arteriole coming in, and this is the glomerular or Bowman capsule or renal corpuscle right there. So this is the glomerulus, but the juxtaglomerular apparatus is right here. You see how closely they are related with each other. Distal convoluted tubule, afferent arteriole, efferent arteriole. The juxtaglomerular cells are basically a smooth muscle cells, which is present in the afferent arterioles. Distal tubule has the macula densa, and this one right here is the mesangial cells. So if there is a hypovolemia or hyponatremia, like low water content, or there's a hypertension, the the distal convoluted cells right here, the mesangials, the macula densa cells, they will signal the juxtaglomerular cells up there to release renin, and it can also signal the juxtaglomerular cells to actually constrict or dilate depending on the situation to control the blood pressure. It will also release the renin to activate the renin angiotensin aldosterone pathway. Another aspect of the histology is the nerve supply to the nephrons and kidneys. So renal innervation include the both afferent and efferent nerves via renal nerve plexus. Innervation is via autonomic nervous system. We have discussed that in detail in sympathetic and parasympathetic nervous system. So if you want to, you can go ahead and look at the neurology slides, the neurology playlist that I have, and you will appreciate it more over there. So the sympathetic efferent nerves, they are primarily through the splanchnic nerves, and the only efferent nerves to the nephron and renal vasculature, and concentrated mostly around the afferent arterioles. Their stimulation can activate the renin angiotensin aldosterone system. So this is important. The signal is given, and it went up to the nerve and the autonomic nervous system. They activate this whole system as well. The sensory afferent nerves are involved in the blood pressure regulation, and visceral afferent nerves will transmit pain to the spinal cord segment T11 to L2. The next part is the ureters. Ureters are the muscular tube which passes urine down to the bladder, and they are lined with the transitional epithelium or the urothelium. So they persist in the ureters, bladder, and to some degree in the urethra. It is called transitional epithelium because it changes its appearance when the bladder and ureters are distended. The next one is the bladder. It is the area for storing the urine, and it is lined with the thick stratified epithelium layer when empty. However, there are plaques on the surface of these epithelial cells when the bladder is empty. And when the bladder is distended, the surface cells are flattened to more cuboidal shape, hence the name transitional epithelium, just like before. And these plaques are important because they prevent the salt and water to permeate through the bladder and back in the body. So they're impermeable and make a seal. There's no point of storing something somewhere if it is leaking. So the plaques are important for that. Finally, we have urethra, male and female. The female urethra is 5 centimeters long and consists of urothelium in the beginning, and then it is lined with the non-keratinized stratified squamous epithelium. And the male urethra is usually 20 centimeters long, and it is divided into prostatic urethra, membranous urethra, and the penile urethra. With this slide, we are going to end our presentation here, and the next time we're going to start learning the physiology of the renal system. Thank you so much, and I'll see you soon.